Welcome to this explainer. Today, we're going to unpack the incredibly complex and, honestly, often mysterious world of primary immune deficiency diseases. I want you to imagine our immune system not just as a random collection of cells, but as this sprawling, heavily fortified castle. When it's functioning perfectly, this fortress is out there repelling countless invaders every single day. But what happens when there's a fundamental flaw in the original blueprint?
Now, you might be wondering, what exactly are primary immune deficiency diseases, or PIDs? Well, they aren't just devastating illnesses. They're actually incredibly precise molecular clues. Because these are inherited genetic defects, they act almost like a magnifying glass. They show us exactly what happens when specific, single components of our fortress defenses break down. By studying where the defense fails, we literally learn how the normal host defense mechanisms are built in the first place. It's like reverse engineering our own immunity.
So, to make sense of all this complexity, we're going to trace the systemic breakdown across 4 key areas. First, we'll look at the defects in our first responders. Then, what happens when T cells fail. Third, the antibody deficiencies. And finally, we'll look at what happens when the regulatory brakes break down completely.
All right, Section 1, Defects in First Responders. Let's head straight to the outermost walls of our fortress and check on the innate immune system. Think of your phagocytic first responders, the neutrophils and macrophages, as your frontline infantry. Well, it turns out they can fail in 3 fundamental ways. First, they might never be produced at all. Take severe congenital neutropenia, for example. It's often caused by a mutation in the ELANE gene affecting neutrophil elastase. These patients have almost 0 neutrophils, and the clinical hallmark is this terrifying absence of pus during severe bacterial infections. Second, in leukocyte adhesion deficiency, the troops are actually produced, but they get totally lost in transit. And third, in chronic granulomatous disease, the troops arrive right on time, but their weapons are completely broken.
When we visualize leukocyte adhesion deficiency, or LAD, it is wild. You can see how those white blood cells are completely trapped inside the red bloodstream. Why? Because they have a genetic defect in their beta-2 endocrines. Those are the little molecules they need to stick to the blood vessel walls. So they're utterly unable to adhere to the endothelium and cross over into the infected tissue. You just end up with these massive amounts of white blood cells circulating uselessly in the blood, all while severe, pus-free infections are just ravaging the soft tissues nearby.
Now flip that around, and you get chronic granulomatous disease, or CGD. Here, the cells arrive at the battlefield looking perfectly fine, but they lack the reactive oxygen species, or ROS, needed to actually kill the engulfed invaders in their phagolysosomes. It's literally like sending infantry into battle with 0 ammunition. Because of this, patients become uniquely susceptible to catalase-positive bacteria like Staphylococcus aureus and filamentous molds like Aspergillus. And because the body can't destroy the invaders, it goes into a panic. It frantically tries to wall them off, building these sterile, microscopic prisons called granulomas. Tragically, these can grow and end up blocking vital organs like the bladder or the bowel. Okay, moving on to Section 2, when T cells fail. We're going deeper inside the walls now to the adaptive immune system, where our highly specialized T cell assassins live.
So T cell development is essentially this incredibly complex assembly line taking place in the bone marrow and thymus. But when genetic roadblocks pop up here, they lead to severe combined immunodeficiency, or SID. This could be due to a toxic buildup from adenosine deaminase, or ADA deficiency, or maybe a lack of crucial survival signals in X-linked SID, or even disrupted T-cell receptor signaling. These blocks halt production entirely. It leaves an infant totally defenseless against practically any pathogen out there, requiring incredibly aggressive therapies like stem cell transplantation just to survive. It's really crucial to contrast the total devastation, the completely empty barracks of SCID, with selective defects like CD3 or ZAP70 deficiency. ZAP70 is this protein that's vital for passing along the signal once a T cell recognizes an enemy. In this specific defect, the CD4 positive helper T cells are physically present in the body, but because their internal alarm bells are broken, they're completely deaf to activation signals. The troops are standing right there on the battlefield, but they simply won't fight.
And you know, it's not just the cells themselves. We see structural and regulatory failures too. Take DiGeorge syndrome. The actual training ground for the T cells, the thymus, is completely missing because of a chromosomal deletion. No training ground means virtually no mature T cells.
Then you have Omen's syndrome, which is fascinating but honestly terrifying. It's caused by a leaky genetic block in the assembly line. A tiny fraction of T cells manage to develop, but their development was so flawed that they become this rogue, hyperactive subset of oligoclonal T cells. Instead of protecting the host, they attack the host's own skin, causing this severe red, scaly rash known as erythroderma.
Right, let's jump into Section 3, the antibody deficiencies. Time to talk about the heavy artillery of our fortress defense, the B cells.
Let's trace a very specific manufacturing defect: X-linked agammaglobulinemia, or XLA. It all starts with one single mutation in the BTK gene, Bruton's tyrosine kinase, located on the X chromosome. Because this kinase is broken, the developing B cell can't send the signal to keep maturing. The entire assembly line gets severely blocked right at the pre-B cell stage. The result? The patient produces virtually zero mature B cells. We're talking less than 1% of normal. That means they have absolutely no heavy artillery to fire at bacterial invaders.
But then you have this bizarre, paradoxical contrast with hyper-IgM syndrome. A patient will actually have normal or sometimes even soaring levels of basic IgM antibodies. But when you look at the IgG and IgA levels, they are dangerously low. It's like the body is mass-producing the default IgM ammunition, but completely failing to upgrade its weapons to the highly specialized, mature IgG and IgA artillery that's so desperately needed for long-term defense. So why on earth does this happen? Well, it boils down to a massive communication breakdown. For a B cell to upgrade from IgM to IgG or IgA, which is a process called class switch recombination, it actually needs direct permission from a T cell. This permission is granted when the CD40 ligand on the T cell binds to the CD40 receptor on the B cell. If a genetic mutation breaks either of those two molecules, the T cells simply cannot give the final order. The B cells are just left stuck in a loop, producing only the basic IgM. Now, as we get older, the fortress can face a totally different kind of threat: common variable immunodeficiency, or CVID. This one is much more mysterious. It's an adult-onset condition where the body basically just stops maintaining adequate levels of antibodies and even fails to produce them when prompted by vaccines. But CVID isn't just an open door to infections. It's a state of profound immune dysregulation. These patients often develop swollen lymph nodes, sterile granulomas, and rogue autoimmunity, where the immune system actually attacks itself. While some cases are linked to specific gene mutations like ICOS or TACI, many are the result of a really complex, still undiscovered constellation of genetic factors. Actually, contrast that severe dysregulation with something like selective IgA deficiency, which is the most common primary immunodeficiency. In that case, only the IgA mucosal defense is missing, often leaving patients completely asymptomatic or maybe just prone to some mild respiratory infections. Finally, we arrive at Section 4, broken brakes and regulatory defects. What exactly happens when the immune system's weapons are working perfectly fine, but the brakes are just gone?
There's this condition called hemophagocytic lymphohistiocytosis, or HLH for short, and its definition is terrifyingly precise. It's an unremitting activation of T cells and macrophages. This is often triggered by something as common as the Epstein-Barr virus, which, by the way, is also the trigger for related regulatory defects like X-linked lymphoproliferative disease, or XLP. But once triggered, it turns into an absolute catastrophic friendly fire nightmare. The defense force completely loses its ability to stand down. It permanently activates and literally starts shredding the host's own organs, particularly the liver, the bone marrow, and the central nervous system.
To spot this runaway train before it crashes, clinicians have to look for immense, massive systemic inflammation markers. We're talking unyielding fevers, massive enlargement of the liver and spleen, and skyrocketing levels of ferritin in the blood. If you look at the bone marrow under a microscope, you will literally see macrophages in a state of absolute overdrive, actively consuming the body's own red and white blood cells. That process is called hemophagocytosis. It is, quite literally, the immune system consuming itself, all because that one genetic off switch is broken. So we've walked through some pretty devastating consequences of what are essentially just tiny genetic typos, from missing troops to broken communication lines to completely destroyed brakes. But I want to leave you with a final provocative thought. If these diseases are built into the very genetic blueprint of our ultimate defense, how do we fix them? Well, the answer lies right at the cutting edge of modern medicine. We are learning to actually replace and rewrite the blueprint itself. Through allogeneic stem cell transplants, and now these revolutionary gene therapies, we aren't just treating infections anymore, we're rebuilding the fortress from the ground up.
Thanks for joining me on this explainer, and keep questioning how our brilliant, incredibly complex bodies work